Okay, so let's dive in. Think about your body as, well, like an incredibly sophisticated chemical processing plant. Every single day, you're exposed to thousands, literally thousands of different chemicals from the world around you, and the air you breathe, the food you eat, even, the products you use. We call these xenobiotics, meaning foreign to the body.
And the sheer volume is truly mind-boggling. But our bodies aren't defenseless. We've evolved this remarkable, highly complex system specifically designed to handle these foreign chemicals, including, critically, the drugs we take, its main job, to transform them. Transform them so they can be safely shown the door.
Shown the door, right. And for this deep dive, we have a stack of sources.
Yeah.
Specifically, it's a deep dive into a chapter all about drug metabolism.
Yeah, exactly. That really lays bare the inner workings of this amazing system. Our mission here is to kind of peel back the layers, understand the key players, and figure out why this isn't just some obscure biochemical process, but something absolutely essential. Essential for both protecting us from toxins and determining if your medicine works, or maybe why it might cause problems. At its heart, the challenge is chemistry. Many foreign chemicals, including most drugs, are what we call lipophilic. They love fat.
Okay, lipophilic.
This means they can easily slip through cell membranes and, build up in our fatty tissues, which is generally not a good thing.
Right. If they just hung around, they could cause all sorts of issues. So the body needs to change them.
Exactly. The ultimate goal of this metabolic system is to make them much more hydrophilic, water-loving.
Okay, water-loving.
Once they're water-soluble, they can dissolve in our body fluids and be easily eliminated, primarily by your kidneys and urine or by your liver and bile.
So this detoxification system is just running constantly.
Nonstop. Consider the sources. Chemicals from plants, whether they're like protective compounds plants make or actual toxins in things like mushrooms, environmental contaminants all around us, food additives, cosmetic ingredients, processed foods. It's a lot.
And what's really fascinating is that the exact same set of enzymes that handles the compounds in your morning coffee or pollutants in the air, they're the ones grappling with your prescription pill.
Exactly. They used to be called drug metabolizing enzymes, but honestly, xenobiotic metabolizing enzymes is definitely the more accurate name.
Because their job is so much broader.
Way broader than just pharmaceuticals. And this system is ancient. It's been shaped by evolution, the differences we see in metabolic enzymes between species, probably adaptations to different diets over millions of years.
Like a chemical arms race.
Sort of, yeah. An ongoing chemical arms race. Plus, these enzymes are also essential for processing and eliminating certain harmful chemicals our own bodies produce. Things like bilirubin, a breakdown product of red blood cells, or excess steroid hormones. Got to deal with those too.
So when you take a drug, you're essentially just adding another xenobiotic into this ancient, constantly working system.
You got it. And it makes sense when you remember that many drugs, as our sources point out, were originally discovered in plants.
Ah, right. The body was already kind of equipped.
It was already dealing with plant chemicals. Now, here's where it gets a little complicated, and honestly, quite challenging for medicine. While this metabolic system is overwhelmingly beneficial, keeping us safe from this chemical soup, its very complexity creates major hurdles for drug development.
Hurdles like what?
Well, we see big species differences, which makes animal testing tricky for predicting human effects. There's enormous variation in how fast or slow different people metabolize drugs, leading to variable responses. And these enzymes, they're ground zero for many potential drug interactions.
And isn't there a risk that this system, it's supposed to protect us, but can it actually create something toxic from something relatively harmless?
Absolutely, and this is a critical point. While metabolism usually inactivates drugs, it can sometimes convert them into highly reactive, unstable intermediates. These are often called electrophilic metabolites. Think of them as little chemical magnets looking for something to stick to.
And what do they stick to?
Crucial cell components like DNA, RNA, or proteins. And this can damage cells, cause organ toxicity. The liver is often hit hard, known as hepatotoxicity, because it's such a metabolic hub.
Right.
And if they damage DNA, they can even lead to mutations that contribute to cancer. This is why rigorous safety testing is absolutely non-negotiable, especially for drugs you might take long-term.
Okay, so how does the body actually do these chemical transformations? Our sources talk about two main stages, right?
Right, phase 1 and phase 2. Think of phase 1 as like putting a handle on the molecule.
A handle.
Yeah, it involves reactions like oxidation, which is adding oxygen, reduction, adding hydrogen or electrons, or hydrolysis using water to break a bond. The goal is to either add or expose a functional group on the drug molecule, like a little chemical flag, maybe an AOH group or an ACOH group.
Okay. And adding that handle doesn't make it super water soluble yet.
Not significantly on its own, no, but it does something else important. It can change the drug's biological activity. Often, this inactivates the drug, stopping its effect. But sometimes, surprisingly, phase 1 reactions activate a drug.
Activate it.
Yeah, this is the concept behind prodrugs. They're given in an inactive form and only become active after metabolism. Examples include some cancer drugs like cyclophosphamide and the blood thinner clopidogrel. Both are activated by these phase 1 enzymes.
Got it. So phase 1 is either preparing the molecule or maybe flipping its activity switch. What happens in phase 2?
Phase 2 is the conjugation phase. This is where the body attaches a much larger, very water-loving molecule to the drug or its phase 1 metabolite. It's like adding a big soluble tag.
A tag, like what?
Common tags include glucuronic acid, sulfate, glutathione, or an acetyl group. These tags are often attached right onto those functional groups that phase 1 just added or exposed.
And that really boosts the water solubility for excretion.
Absolutely. This is the step that dramatically increases water solubility, making it easy for the kidneys or liver to eliminate the compound. Like phase 1, phase 2 usually turns off biological activity, but again, there are exceptions. Yeah, where the conjugated form of a drug can actually be more active than the parent, like certain forms of morphine or minoxidil. It's not the rule, but it happens.
So phase 1 often sets the stage for phase 2, but a drug could potentially skip phase 1 and go straight to phase 2.
Exactly. And both phases rely on large families or superfamilies of enzymes.
Okay, who are they?
For phase 1, the big names are the CYPs, that's cytochrome P450s, FMOs, flavin-containing monoxygenases, and epoxide hydrolases, or EHs. For phase 2, you'll hear about GSTs, glutathione S transferases, UDP, glucuronidyltransferases, SALTs, sulfotransferases, NATs, and acetyltransferases, and MTs, methyltransferases.
Wow, that's a lot of acronyms.
It is. A general rule, though, is that phase 2 enzymes tend to work faster, catalytically speaking, than phase 1 enzymes.
Okay, so where in your body are these chemical transformations actually taking place? Where's the action happening?
These enzymes are found in most tissues to some extent, but the absolute highest concentrations, the real metabolic powerhouses, are in the gastrointestinal tract.
The GI tract.
Yep, especially the liver and the small and large intestines.
Okay, let's trace the path of a drug you swallow. What happens first?
First stop, your gut bacteria. They have their own metabolic enzymes and can start working on the drug right away. Then, as the drug is absorbed through the intestinal wall, it encounters enzymes right there in the intestinal cells. That's an initial metabolic hit.
Okay, even before it gets into the blood properly.
Exactly. From the intestines, absorbed drugs travel via the portal vein directly to the liver.
And this is the famous first pass effect.
Precisely. The liver is the primary metabolic clearinghouse. A significant amount of the drug can be metabolized here on its very first pass, before it even gets into the main bloodstream to circulate throughout your body.
So it gets knocked down before it even gets going.
Sometimes quite a bit, yeah. Drugs that escape this first pass will be metabolized further each time they circulate back through the liver until they're eliminated. If a drug is metabolized very slowly, well, it'll stay in your system longer, leading to a longer half-life.
Are there other important sites besides the gut and liver?
Yes, definitely. Tissues like the nasal passages and lungs also have significant metabolic activity. That's particularly relevant for things like inhaled drugs or, air pollutants.
And inside the cell itself, where are these enzymes hanging out? Are they just floating around?
Good question. Many of the phase 1 enzymes, like most CYPs, FMOs, and one type of epoxide hydrolase called MEH, plus some phase 2 enzymes like UGTs, are actually embedded in the membrane of the endoplasmic reticulum, the ER.
The ER membrane, why there?
Well, it's a perfect spot for interacting with those fatty hydrophobic drugs that easily move into cell membranes.
Makes sense. And the others?
Other phase 2 enzymes like GSTs, SALTs, NATs, MTs, and another type of epoxide hydrolase called SEH are found floating in the cytosol, which is the main fluid part of the cell.
All right, let's put some names to faces, metabolically speaking. Who are the real stars of phase 1?
You absolutely have to start with the CYPs, the cytochrome P450 superfamily. These are the largest and by far the most studied enzyme family in drug metabolism. They contain an iron-containing structure called hemum, and they use oxygen and a molecule called NADPH to perform a huge variety of oxidation reactions.
How many of these CYP enzymes are there? You said a superfamily.
There are over 50 in humans. But here's a surprising point. The metabolism of the vast majority of clinical drugs we're talking on, most of the pills you might take is handled by just a small group.
Really? Only a few?
Yeah, maybe about 15 human CYPs are really important, with around 12 being the heavy hitters for foreign compounds for xenobiotics.
You mentioned earlier that one of them, CYP3A4, is responsible for metabolizing over half of all drugs. That's huge.
That's right. CYP3A4 is the absolute kingpin in the liver, responsible for over 50% of the drugs prescribed today. It's incredibly important. And you also hit on something else key. These drug-metabolizing CYPs are rather promiscuous.
Promiscuous, like they'll metabolize anything.
Not quite anything, but they can process many structurally different compounds, and they can often attach that handle at multiple different positions on the same molecule.
How can they do that?
It's because they often have relatively large and flexible active sites compared to, say, enzymes that only work on one specific molecule in your body's core metabolism. Think of it like a flexible lock that different keys can fit into.
Okay, so they're flexible. But how does that flexibility lead to drug interactions? You mentioned that earlier.
Well, that flexibility means different drugs can often fit into the active site of the same CYP enzyme.
Ah, so they compete for the enzyme.
Exactly. Leads to competition.
Yeah.
If you take two drugs metabolized by the same CYP, they compete for enzyme time, potentially slowing down the metabolism of one or both, which can lead to problems.
Right, but competition isn't the only way, is it? You mentioned inhibition and induction, too.
Correct. Drugs can also inhibit a CYP, directly blocking its activity, or they can induce a CYP, which means making your body produce more of the enzyme protein.
Okay, so let's unpack inhibition. If one drug inhibits a CYP that metabolizes another drug, the second drug could build up to toxic levels.
Precisely. A classic example is the antifungal drug ketoconazole. It's a potent inhibitor of CYP3A4. So if you take ketoconazole along with another drug that's primarily cleared by CYP3A4, the inhibited enzyme can't do its job properly. The second drug's levels can soar, potentially leading to toxicity.
Wow, okay. And induction is the opposite, making more enzymes.
Yes. Things like certain steroid hormones, or even the herbal supplement St. John's wort, which contains a compound called hyperforin, can cause your body to make significantly more CYP3A4.
So what happens then?
Well, if you then take a drug metabolized by CYP3A4, it gets cleared out much faster than usual, potentially making it less effective.
Like with birth control.
Exactly. This is why St. John's wort can reduce the effectiveness of things like oral contraceptives or certain blood thinners. Even dietary stuff matters. Compounds in grapefruit juice are well-known inhibitors of CYP3A4.
Grapefruit juice. The story of the old antihistamine, terfenadine, really hammered home the dangers of these CYP interactions, didn't it?
It really did. Terfenadine itself was actually a prodrug, metabolized by CYP3A4 to its active form. That was fine. However, if CYP3A4 was inhibited by other drugs or even grapefruit juice, the parent terfenadine, the inactive form, would accumulate to high levels.
That was bad.
Very bad. This parent compound caused dangerous heart rhythm problems, cardiotoxicity, which ultimately led to its withdrawal from the market. It was a huge lesson in predicting and managing metabolic interactions.
A really stark lesson. And it's not just interactions with other substances. Our own genetic differences play a massive role with CYPs too, right?
Absolutely massive. Genetic variations, called polymorphisms, in certain CYP genes, particularly CYP2D6, are very common in the population. Some people have versions of the gene that make their CYP2D6 work very slowly. We call them poor metabolizers. Others have versions that make it work super fast, ultra-rapid metabolizers.
And CYP2D6 handles a lot of drugs.
A significant number of common drugs, yes, certain antidepressants, beta blockers, many pain relievers like codeine. So these genetic differences can dramatically affect how you respond to a drug. It might require big dose adjustments, or it could lead to treatment failures or unexpected toxicity.
And the point where some drugs have been pulled.
Yes, this variability has actually led to some drugs being taken off the market if their safety window was too narrow and too dependent on this highly variable enzyme.
Okay, so CYPs are clearly the big players. But beyond the famous CYPs, who else matters in phase 1?
We should definitely mention the flavin-containing monoxygenases. the FMOs. They're also in the ER membrane, especially in the liver. FMO3 is the main one in the adult human liver and handles things like nicotine and certain drugs containing nitrogen or sulfur.
Okay. Is FMO3 linked to any conditions?
Interestingly, yes. If you have a severe genetic deficiency in FMO3, you can develop a condition called trimethylaminiuria or fish odor syndrome.
Fish odor syndrome.
Yeah, an unmetabolized dietary compound, trimethylamine, builds up and is excreted, causing a strong fishy body odor. It highlights FMO3's role in handling specific dietary components.
Are FMOs as big a player in drug interactions as CYPs?
Generally, no. They tend to be less involved in the metabolism of the most commonly prescribed drugs compared to the major CYPs. And importantly, they seem to be less prone to being inhibited or induced by other drugs or those nuclear receptor switches that regulate CYPs.
So that makes them potentially better targets for new drugs.
Exactly. It actually makes them an attractive pathway to aim for when designing new drugs if you want less variable metabolism and fewer interaction worries.
Interesting strategy. And then there are the hydrolytic enzymes you mentioned, using water to break bonds.
Yes, key among these are the epoxide hydrolases, the EHs. Now, CYPs often produce epoxides as part of their reactions.
And epoxides can be bad news, right? Those reactive sticky things.
Many of them can be, yes. Potential toxins or carcinogens. So EHs hydrolyze these epoxides, essentially neutralizing them. They play a crucial protective role. There are two main types relevant here. One in the ER membrane, called MEH for microsomal, and one in the cytosol, called SCH for soluble.
Can you give an example of MEH in action? Detoxifying something?
Sure. The anti-epileptic drug carbamazepine is first metabolized by a CYP into an active epoxide form. Then MEH comes in and hydrolyzes that epoxide, inactivating it. But if you were to take another drug that happens to inhibit MEH, the active carbamazepine epoxide could build up, potentially increasing side effects.
So it shows how important the whole metabolic chain is.
Exactly. You have to understand all the steps.
Okay, now here's where our sources got really interesting. Tell us more about that soluble epoxide hydrolase, SEH. You said it handles internal molecules too.
Yes, the soluble EH, SCH, has garnered a lot of attention recently. Its substrates aren't just foreign chemicals, but also some important endogenous signaling molecules. Specifically, it metabolizes certain fatty acid epoxides. These are derived from essential fatty acids like arachidonic acid and also those omega-3s and omega-6s you hear about.
And these fatty acid epoxides, they do good things.
Well, in experimental systems, they've been found to have beneficial properties. They can be anti-inflammatory, reduce certain types of pain, and even help lower blood pressure.
Okay, so if SEH breaks down these beneficial compounds, the logical step is maybe we should inhibit SEH.
Exactly. That's the thinking. If you inhibit SEH, those beneficial fatty acid epoxides stick around longer, potentially boosting their positive effects like reducing inflammation or pain.
So it's using the body's own pathways.
Precisely. It's a prime example of how understanding the body's own metabolic pathways can lead to new drug targets. Companies are actively exploring SEH inhibitors right now as potential new therapies.
Fascinating. And quickly, carboxylesterases, another hydrolysis enzyme.
Yes, another superfamily using hydrolysis, breaking down esters and amides. They're found in various cellular locations. They're involved in detoxifying many compounds, but also, importantly, in activating some prodrugs.
Like that cancer drug you mentioned.
Right. A key example from our sources is the cancer drug oronodekin. It's given as a prodrug, and it's activated by carboxylesterase 2, or a CES2, into its potent chemotherapy form called SN38.
Okay. All right, shifting gears to phase 2, the conjugators. Adding those big tags. Who are the heavy hitters here?
The UGTs, UDP glucuronidyltransferases, are definitely major league players. They attach glucuronic acid, a sugar derivative, to a huge variety of substances, not just drugs, but also crucial endogenous compounds like steroid hormones, bile acids, and very importantly, bilirubin.
Bilirubin again?
Yep, and like the phase 1 enzymes, UGTs are highly concentrated in the gut and liver, making them absolutely crucial for that first-pass metabolism effect we talked about.
And the goal of adding that glucuronic acid tag is elimination, making it water-soluble.
Primarily, yes. The glucuronides, the tagged molecules, are typically much more water-soluble and easily eliminated, usually in urine or, for larger conjugates, in bile. But here's something fascinating about the bile route.
Oh.
Glucuronides excreted in bile go down into the large intestine. And the bacteria living there, our gut microbiome, have enzymes called beta-glucuronidase. These bacterial enzymes can actually cleave off that glucuronic acid tag, releasing the original molecule, back into the gut lumen.
No way, so it gets untagged.
Exactly. And if that molecule is still capable of being absorbed, it can get back into the bloodstream and circulate all over again.
So it's like a metabolic boomerang.
That's a great way to put it. It's called enterohepatic recirculation, and it can significantly extend the time a drug or its metabolite stays in the body.
Like with that chemotherapy metabolite, SN38?
Precisely. Its glucuronide metabolite undergoes this recirculation. Active SN38 gets released back into the gut, which is a major reason why it can cause such significant gut toxicity like severe diarrhea.
Wow. How clinically important are UGTs for us, for the listener, beyond that recirculation effect?
Extremely important. Perhaps the single most powerful example involves UGT1A1. This specific UGT enzyme is absolutely essential for clearing bilirubin.
That yellow pigment from red blood cell breakdown.
Right. And get this. UGT1A1 is the only xenobiotic metabolism enzyme that is known to be essential for human life. Your body must be able to get rid of bilirubin constantly.
What happens if it doesn't work?
If UGT1A1 isn't working properly, bilirubin builds up, causing hyperbilirubinemia, which we see visually as jaundice, that yellowing of the skin and eyes.
Like in newborns.
Exactly. Many newborns have temporary jaundice, partly because their UGT1A1 system isn't fully mature yet.
And there are genetic conditions linked specifically to UGT1A1.
Yes. Very severe mutations cause rare but life-threatening conditions called Crigler-Najjar syndromes, where bilirubin clearance is almost non-existent. But much, much more common is Gilbert syndrome. This is a common, generally benign condition found in maybe 8 to 23% of the population, depending on ancestry. It just causes slightly elevated bilirubin levels.
And what causes Gilbert syndrome?
It's often due to a common genetic variation in the promoter region of the UGT1A1 gene, the part that controls how much enzyme gets made. This variant, called UGT1A128, just means your body makes a little less of the UGT1A1 enzyme.
Okay, slightly less enzyme. How does having Gilbert syndrome affect drug metabolism then?
Because you have less UGT1A1 enzyme activity, you can be significantly more sensitive to drugs that are heavily metabolized by UGT1A1.
Like irinotecan again.
Exactly. That active chemometabolite, SM38, it's primarily inactivated by UGT1A1 and a related enzyme. In someone with reduced UGT1A1 activity from Gilbert syndrome, SM38 isn't cleared as efficiently. Add in that enterohepatic recirculation loop we talked about, and you get higher levels of toxic SM38,
Right.
particularly in the gut.
Leading to worse side effects.
Much worse. Severe, sometimes life-threatening diarrhea and blood-related toxicities. It's a prime example where knowing your UGT1A1 genotype can be absolutely crucial for safe prescribing and dosing.
And some drugs can inhibit UGT1A1 too, right? Not just rely on it.
Yes, exactly. The HIV drug atazanavir is a known inhibitor of UGT1A1. So in patients with HIV who also have Gilbert syndrome, taking atazanavir can lead to significantly higher bilirubin levels because both their underlying genetics and the drug are hitting that same UGT1A1 pathway.
Okay, so UGTs are huge. Who else is a major phase 2 player?
The GSTs, glutathione S-transferases, these enzymes play a really key defensive role. They attach glutathione, which is a small molecule abundant in our cells, to those reactive electrophiles we keep mentioning, the sticky molecules that can damage DNA, proteins, etc.
So they're like chemical mops.
Pretty much. By attaching glutathione, GSTs essentially mop up these reactive toxins before they can do damage. They're highly concentrated in cells as a first line of defense.
And like CYPs and UGTs, are there common genetic variations in GSTs that affect people?
Yes, definitely. There are well-known variations where individuals can completely lack the activity of certain GST enzymes, like GSTM1 or GSTT1. These are called null phenotypes.
And what's the consequence of lacking one of these GSTs?
Well, having these null genotypes has been linked to increased susceptibility to toxicity from specific environmental exposures or certain drugs. And interestingly, it's also been associated with an increased risk for some types of cancer, like lung, colon, and bladder cancer, likely because the body is less efficient at clearing certain carcinogens without that specific GST activity.
But you mentioned a paradox earlier with GSTs in cancer treatment, something about resistance.
Yes, it's a fascinating twist. While GSTs protect normal cells from toxins, cancer cells can sometimes hijack the system. They can overexpress GSTs, making them more resistant to chemotherapy.
How?
They basically use the GSTs to quickly detoxify the very chemotherapy drugs we're giving to try and kill them.
Wow. So they use our defense against us.
They do. But this has also opened up new therapeutic avenues. One strategy is to develop GST inhibitors to give alongside chemotherapy, making the cancer cells more vulnerable again. Another really clever approach is to design prodrugs that are activated specifically by the high levels of GSTs found only in the tumor cells.
So you turn the cancer's defense into its own poison delivery system.
Exactly. Using the cancer's own upregulated GSTs to deliver a targeted toxin right where it's needed.
That's really smart.
Yeah.
Okay, what about the NATs? N-acetyltransferase.
Right, the NATs. They add an acetyl group to drugs or chemicals, particularly those with specific structures like aromatic amines or hydrazines. Adding this group can sometimes make the molecule less water-soluble, actually, but it often neutralizes a potential charge, changing its properties and usually inactivating it.
And these have a historical significance.
They really do. If we connect this back to the history of pharmacology, the characterization of acetylator phenotypes, figuring out whether someone was a slow or rapid acetylator based on how they metabolized the tuberculosis drug isoniazid. That was one of the very first times scientists clearly demonstrated that a person's specific genetic makeup could dramatically alter their response to a drug.
Wow, so this was foundational for pharmacokinetics.
Absolutely foundational. It showed really clearly that drug response isn't uniform across the population. There are two main NAT genes in humans, NAT1 and NAT2. The slow acetylator status, which is quite common, is primarily linked to variations in the NAT2 gene.
And what happens if you're a slow acetylator?
You clear NAT-dependent drugs more slowly. This includes things like the blood pressure medication hydralazine or certain antibiotics called sulfonamides. Slower clearance means higher drug levels and an increased risk of concentration-dependent side effects.
And there's a link to cancer risk with NAT2 slow acetylators, too, something about the bladder.
Yes, particularly bladder cancer risk, especially when associated with exposure to certain environmental chemicals like bicyclic aromatic amines found in things like cigarette smoke or some industrial processes.
How does that work?
Well, fast acetylators can often detoxify these compounds efficiently in the liver via NAT2. But in slow NAT2 acetylators, these compounds can accumulate, get excreted in urine, and then reach the bladder lining. In the bladder epithelium, a different NAT enzyme, NAT1, is highly expressed. NAT1 can perform a specific metabolic step called N-hydroxyacetylation on these accumulated compounds. This pathway, unfortunately, creates highly reactive DNA-damaging intermediates right there in the bladder tissue, increasing the risk of bladder cancer, especially in smokers who are also slow NAT2 acetylators.
That's a really specific and dangerous interaction.
Right.
Okay, finally, just briefly on SALTs and MTs.
Right. SALTs are sulfotransferases. They add a sulfate group. MTs are methyltransferases, adding a methyl group. Both steps generally modify the molecule, often increasing solubility or terminating biological activity.
Is there a key clinical example for those?
A really crucial one involves MTs, specifically an enzyme called thiopurine methyltransferase, or TPMT. TPMT metabolizes a class of drugs called thiopurines, like azathioprine or 6-mercaptopurine. These are used for conditions ranging from autoimmune diseases to childhood leukemia.
And what's the issue with TPMT?
Individuals with genetic deficiencies in TPMT activity, which occurs in a small but significant portion of the population, are at extremely high risk of severe, potentially fatal bone marrow toxicity from standard doses of these drugs.
Why?
Because the toxic drug metabolites build up rapidly if TPMT isn't there to break them down.
So testing is important.
Absolutely critical. Genetic testing for TPMT deficiency is now standard practice before starting thiopurine therapy. It allows doctors to identify patients at risk and use significantly lower doses or choose alternative drugs altogether to avoid these devastating side effects. It's a major success story for personalized medicine.
Okay, so bring this all together. Metabolism is obviously complex.
Yeah.
How fundamentally does understanding this whole system impact how we use drugs safely and effectively today?
Oh, it's everything. Absolutely fundamental. This system determines how long a drug stays in your body, its half-life. If your system clears a drug too quickly before it has a chance to work, it might not be effective. If it clears it too slowly, the drug builds up, leading to side effects and potential toxicity.
And adverse reactions are a big problem.
A huge problem. Adverse drug reactions, ADRs, are a major cause of hospitalizations and significant harm, and a massive percentage of them are directly related to how an individual person metabolizes drugs. And that metabolism, as we've seen, is influenced by their genetics, what they eat, their environment, other medications they're taking. It's multifactorial.
Which is why drug companies spend so much time and money studying metabolism when they're developing new medicines.
Precisely. It's a mandatory part of development. Before any new drug gets close to approval, developers have to thoroughly map out how it's metabolized. They need to identify all the breakdown products or metabolites, assess if any of them are active or potentially toxic, and crucially, predict potential interactions with other common drugs via inhibition or induction.
And are they trying to design drugs differently now, knowing all this?
Yes, there's definitely a conscious effort now to design new drug candidates to avoid being primarily metabolized by just one highly variable enzyme like CYP2D6 or even the workhorse CYP3A4.
Why? To minimize problems.
Exactly. If a drug is metabolized by multiple enzymes or by less variable pathways, like maybe the FMOs, it tends to lead to more predictable responses across the population and fewer severe interaction issues.
And how does the concept of induction, where substances make your body produce more enzymes, fit into predicting all this variability?
Induction is another major variable that needs to be understood. Many drugs and chemicals can activate cellular switches called nuclear receptors. When activated, these nuclear receptors travel to the cell's nucleus and basically turn up the production line for specific metabolic enzymes and also drug transporters.
Okay, so these receptors are like master regulators.
In many ways, yes. Key examples from our sources include nuclear receptors like PXR and CAR. These are activated by a really wide range of drugs, things like antibiotics like rifampicin, statins, anticonvulsants, even St. John's wort's hyperforin. And activating PXR or CIR powerfully induces CYP3A4 and other enzymes involved in clearing those substances.
And there are others.
Yeah, another one is the AHR, the aryl hydrocarbon receptor. It's famously activated by environmental pollutants like dioxins and PCBs, but also by components of cigarette smoke and some drugs like the heartburn medication omeprazole. Activating AHR induces CYP1A1 and CYP1A2.
So taking one drug can literally make your body clear another drug faster because it's flipped a switch to manufacture more of the enzyme.
Exactly that. Take PXR induction by rifampicin or St. John's wort. Both strongly activate PXR, which tells your liver cells to pump out significantly more CYP3A4 enzyme protein. So if you're taking, say, oral contraceptives, which are primarily metabolized by CYP3A4, while you're also taking rifampicin or St. John's wort,
the contraceptive gets cleared much faster.
Much faster, potentially leading to reduced effectiveness and unintended pregnancy. It's a classic clinically important induction interaction.
And you mentioned these nuclear receptors can differ between species too, which complicates animal testing again.
They absolutely can. A drug might strongly activate the human PXR switch but barely touch the mouse or rat version or vice versa. This makes it really hard to rely solely on animal studies to predict human induction risks accurately. More hurdles for development.
Okay. Looking ahead then, how is our growing understanding of this whole metabolic landscape shaping the future of drug discovery and actual therapy for patients?
It's profoundly influencing it. Modern drug development involves extensive testing of drug candidates with human liver preparations, human enzymes, cells engineered with specific human nuclear receptors right from the very early stages. The goal, as we said, is to design drugs that are metabolized predictably, ideally by multiple pathways, to make them less sensitive to variations in a single enzyme or prone to major induction issues. Safety and predictability are key.
And beyond just designing better drugs, what's on the horizon for understanding individual responses?
Well, computational prediction, trying to predict metabolism using computer models, is being worked on constantly, but it's still really tough because these enzymes, especially the CYPs, are so flexible and their interactions are complex. But here's where it gets really exciting for the future. The field of metabolomics.
Metabolomics, what's that exactly?
It's not just looking at the drug itself or one or two metabolites. Metabolomics uses advanced analytical techniques like mass spectrometry to measure all the thousands of small molecule metabolites present in a biological sample: urine, blood, tissue.
Like a complete biochemical snapshot.
Exactly. A snapshot of what's happening chemically inside you at that moment.
And how's that help with drugs?
It's proving to be a really powerful screening tool. For instance, in preclinical animal studies, researchers can look for specific patterns or fingerprints of metabolites that show up only when a drug is causing toxicity in a particular organ, say the liver or kidney. This could potentially identify safety problems much earlier and more sensitively than traditional methods like waiting to see tissue damage under a microscope.
And in humans?
In clinical trials, identifying specific metabolic fingerprints might help predict which patients are likely to respond well to a drug versus those who won't, or who might be predisposed to experiencing certain toxicities. It gives us a much richer, more holistic picture of the body's overall biochemical response to a drug, beyond just measuring the drug level itself.
Wow. So
That's
it really sounds like our bodies have this incredibly sophisticated, constantly adapting system of enzymes that are, literally fighting battles with the chemical world around us every second.
Yeah.
And that ongoing fight dictates so much about our health and critically how medicines work or don't work for us.
That's a great way to put it. It truly is this dynamic interplay between the constant barrage of chemicals we encounter, external and internal, and our ancient evolved enzyme machinery. And it's all influenced by our unique genetic code, our diet, our environment, and any other drugs we happen to be taking. It protects us, absolutely, but it also introduces this incredible variability and complexity into drug therapy that we're still working hard to fully understand and manage.
And there's a final thought for you, the listener. Just consider this. Building on everything we've discussed today, the dramatic effects of individual genetic differences in enzymes like UGT1A1, NEET-T2, TPMT, and the GSTs. The intricate web of drug interactions mediated by CYPs and those nuclear receptors, and the promise of metabolomics to map your personal biochemical landscape. Is the future of drug therapy destined to become truly personalized? Could medicine one day tailor your treatment plan, predicting efficacy and toxicity based on your unique individual metabolic fingerprint? Or perhaps the deeper insight is simply the sheer, quiet miracle of this hidden enzymatic world constantly defending you, an evolutionary masterclass playing out silently beneath the surface every single day. Something to think about.